forgot to turn on my microphone. But you can hear me, right? Okay. Okay, so let's compute similarity, cosine similarity between two vectors. So what's the equation supposed to be? So I have this x1, x2, two vectors. Based on the equation, so you can compute the cosine theta between these two vectors by using the dot products between them. So I have x1, x2, so you have inner product, which also dot product between these two, then divide by their respective norms product. So what's the equation? What's the expression here? You can do transpose this times x2. So this is the dot product, the 20. You can also use the function dot, so you don't have to worry about how to transpose one of them. So this is the formula which on the top. So you have x1 times x2 vector in the inner product. Then you have a norm of x1 times norm x2 in the denominator here. So this will be your cosine theta. This is a similarity between these two vectors. So in our random example, these two vectors have a cosine theta similarity 0.62. So my x1 is 0. My x1 is this direction. If I have an orthogonal vector, for example, if my x2 is 0, 3, 5, 0, then this will be 90 degree, and this cosine should be 0. If you replace x2 with 0, 3, then you compute cosine theta between them, you should get 0. If x2 is 0, 3, then I compute this. So this tell me these two vectors goes to the different direction, but not opposite. So if you have -3, 0, so it's go completely opposite direction as a x1, then you should have a -1. If -2, so this is -3, 3, 0, and then you distance, percent similarity. So if you go -3, 0, okay? So this equation you can also rewrite into x1 divided by x1 norm times x2 divided by x2 norm. Then you take dot product between them. So you can normalize first. This should be the same if you get this. So if I do x1 divided by norm x1 itself, then you should get the same. So so if you do this way, so basically you put all the vectors back to unit circle first. So you push this back. Then you compute the dot product. They're the same. So if you want to compute the distance, you just have 1 minus it. That's a cosine distance between two vectors. And our example vectors they all located on the x, y axis, but the general rule just apply to any vectors. It doesn't have to be here. So wherever you are, how long, it doesn't matter, okay? So let's see how can we use this information if I have real data. How can we compute? Our real data is big X, remember? So we have genes, cells. And we need some numbers, so I just 5, 0, 1, 2. Let's just make it up. So this 1, 3, 0, 1. Okay, I just made it up. By the way, how I made it up is that we will discuss next week, because I'm not randomly right. I have some idea why I'm writing 5 here and there. So the number pop up from my mind based on some kind of distribution. It's not purely random. I want to make this gene expression on average lower and more zeros, and I want to make this more uniformly. Every gene cell have expression, but not very high. So somehow, so this will be discussed next week about generation of the data. But let's just play with this matrix. If you type this in there, so we can see how can we compute the distance between cells. Okay. Remember, this will be the vector x1. This will be vector x2. We have 5. Okay. Okay, now your job is to compute the pairwise cosine similarity between cells. You have 5 cells. What you have to do is to take the first column, the first cell, compare with the second cell to compute cosine distance or similarity between them. Then you compute 1, 3, 1, 4, 1, 5. So pairwise difference by column. And we are not talking about the genes, but you certainly can compute the similarity between genes. If you have 2 genes, they all express at a similar level in the cells you sampled, then they have high similarity. So you can do by rows, but right now let's try it by columns. How can we write a function or write some code in order to compute the cosine similarity between cells? First, remember, we are going to compute a pairwise distance matrix D. So this D depends on how many cells in there. So these are 5 cells. This D will be 5 by 5. Okay. So this matrix contains the pairwise distance or similarity between each pairs. These all zeros, so we don't have to compute. They are the distance. Let's see, similar. Let's compute the distance. So if you compute the similarity and then you use 1, just minus it. Or I think we just take a negative. Just compute negative cosine theta between I and j. So you need to write a loop. Basically, you will compute this pair. This is 1, 2, and then 1, 3, 1, 4, and put the values in there using the index then you don't have to compute this because 1, 2, and 2, 1 are the same. So let me show you how to do it. So let's first define containers. We need a big D, so this D will be contains all zeros first, the 5 by 5. Okay, so we create a squared matrix, which is D, to hold the results. We are ready. Now, if you you do a D and then 1, 2, that's this value here. 1, 2 is 0 because we haven't done any computation, so we just hold the place. So now if you do D 1, 2, so this is 0, right? So we will extract the first element from X, so the first row, first column, and then our second so I extract 2 columns in order to compare. So my first D 1 and 2 is x1 dot x1 x2 divided by norm times x2. Because it's the distance instead of similarity, so that's why I'm doing this negative sign, so in order to give distance rather than similarity. Okay, so you know how to do this. This is a equation. We have to compute the cosine similarity, and we just replace the index with 1, 2, 2 vectors and give the results to that particular element. So in this, we completed the first step for the computation. So now the D has been updated, and the value of the computation has been assigned to this. So these 2 cells, these 2 first pair of cells have a negative 0.9. This is distance. I would say they are very, very similar if they have a negative distance between them. Okay. So now you have to do the second one between 1, 3. This will be, I replace this X by 1. Okay, so this is a way to compute the second. So you have to just do a loop and to fill in all the 1, 2, 1, 3, 1, 4, 1, 5, and the 2, 2, 3, 2, 5, something like that. So this is a double loop in order to complete this. Okay, so you get the idea. So if you want to replace, so now we compute the cosine distance. You can also compute the Euclidean distance, which is easier. So 1 minus 2 vectors difference, and then you take a norm. The formula will be even easier than this. So if you compute the Euclidean distance, we can use x1 minus x2 norm. So that's all. So the difference between distance between these two is 2.6. What I'm writing on this board here is this one. But I just replace I and j with 1, 2. Then you have to iterate the sub-index. So in order to loop all the pairwise cells. In order to compute the distance. So now I'm going to use Euclidean distance as an example to do this double loop. It will be the same. So this is easy to write. So I will rather not to write this long formula, but use this shorter formula to compute the pairwise distance and fill in this distance matrix. Okay, but you can compute the cosine distance using this. So now I'm going to fill in. So now this is a D, but I will using D. So using minus x2. So I have a pre-computed distance of cosine distance, but now I'm going to replace with Euclidean distance only because this expression is shorter, so easy to demonstrate. So this will replace this value using Euclidean distance between these two cells. Now we can do the 1, 3 and just replace. As you can see, we just iterate the subscript and to get this filled up. It's 1, 4, and then put this. So we are gradually put the distance between cells into this D matrix. So instead of doing this manually 1 by 1, I just do a index using 1 to 5. So this will be 1, 2, 3, 4, 5, and J 1, 2, 3, 4, 5. And each time, and the J will be replaced. And then in this way, you have a double loop to compute the pairwise distance. You don't actually to have compute every combinations. For example, the 1, 1 place, you don't have to compute because they are automatically should be filled with 0. And you shouldn't compute 2, 1 after compute 1, 2. But in order just to save, to make this simple, I just compute every element by itself. And it's not highly efficient, but just very clear. And you do a loop for the I, first I, then 1 to 5J, and then come back again. So this just replace what I just typed in 1, 2, 1, 3, 1, 4. And the result of this should be a D matrix. By the way, you can click this to see the matrix. Okay. So this piece of code will give you the pairwise matrix. You can see the symmetric. The distance from 1 to 2 are the same as 2 to 1. Looks like the distance, the largest is between number 4 and the number 5. So the distance is 9.4. 9.4. So that's these two cells looks the most different from each other. I don't know if you can see from the profile, but anyway, so these two will be the most distinct cells. Which two are the most similar to each other? So 1, 2, these two cells are the most similar, close to each other. But this is based on Euclidean distance. Any questions for this so far? Okay. Yeah. I don't know, but based on the result of computation, the D, if you D12 is 2.6. The number is smaller than other numbers. That's why I say these two cells are the most similar based on the Euclidean distance measurement. And these two cells distance is the most different. That's 4, 5. That's 9.4. So that's what we observed. Yeah. So now with this map, just consider this as a distance between cities. You should be able to rearrange these 5 cells in the space in the 2D map. Okay. So their position is defined by 1, 2, 3, 4, 5 genes. So basically each cell is defined in a 5D hyperdimension space, right? It's defined by 5 numbers. So if you go 0 first, 5 this direction, and then in third dimension you go 0, and then fourth dimension you go 1, and the fifth position you go 2. So the first cell will go somewhere there. And the second cell you go first dimension 1, second dimension 3, and 0, 0, and 2. So the second cell in the fifth dimension goes somewhere there, and then they're separated, scattered in this hyperdimension. But if you want reconstruct a 2D map, the map, you will map 5 cells in the 2D space on this board. You can use this distance relationship. I will put cell first one, x1 here, x2 is close to it, so x2 may be here. And then my 5, 4, I know 4 is far away from x5, so this distance is far. So I can put them into a 2D, let them relationship follow this distance map. So in this way, if I somehow 3, 4, 5 vectors I put in the 2D and reduce the dimension from 5D into 2D and keep their relative distance position. So we are doing a dimensionality reduction from this high dimension representation, original, to this 2D. Any questions? The concept questions? A break, okay? 5 minutes. Okay, the concept is like this. So we obtained expression matrix from a single cell study. It's a big X with 5 cells defined by 5 genes expression each. Then we compute the pairwise distance between each pair of the cell. So in our case, by column. And to fill up this pairwise distance matrix, a D, that's what we have, by looping through every pair in the expression matrix, each pair of cells. Now you know how to do this manually, right? So just do this, fill this up. That's the results we have. Now we are going to use multi-dimensional scaling. So to convert this into a map, each point, each city on the map is a cell. What we want to make a connection between this distance matrix and the map is to keep the relative distance between cells, satisfy this distance matrix. Okay. You can draw this if you put x1, x2 by that amount, then 1 by 3 somewhere. So if you try this, I put x1, x2 together and close this 2 and then try this longer x3. And then I have to make sure x3 and x4 is 5, so not too far away. So you need some arrangement, and it's not so easy. So that's why we are using this function. So if you use multi-scaling fact, so the function is this. So this MDS, multidimensional scaling function, is a built-in function. It needs a distance matrix, the D. So we will give the D to it. And also, you can see, okay, I want a 2D map. The 2D. If you put a 3, that will give you a 3D map. Okay. So this function will return the coordinates of the projection. It will give you the position of x1, x2. So we are going to put that into a figure and then see what the result is. You may receive some warning message because there's not enough information to compute. There's some numeric warning, but it doesn't matter. So I will get x, y coordinates. That's what I get. So the first row is -2, -0.3. That's the location of the first cell, x1. And second cell position should be -0.7 and 1. So you have second cell. So I can plot them in a plot. So I'm going to do this. I have 5 files. So 1, 2, 5 cells. I will plot the x1, the first this is just plotted dots. Yeah, so this is this 5 cells on this map, this location. And if you measure the distance between them, let's see, so this is a number 4. Number 4, where's number 5? Number 5 is here. Number 4, number 5 really far away to each other. Number 4, 4, 2. Number 4, that's number 3. 4 and 3. So this will be the multi-scaling, dimension scaling, the results. So the concept here is you have high dimensional data, 5 cells defined by 5 genes. Or this can be longer, 5,000 or more. But our small example, a hand drawing, is just 5 genes. But anyway, so you can find the similarity, this similarity between cells to compress the information into a distance matrix. That distance matrix can be projected to let them to project into a 2D space and keep the relationship between cells. Okay. So we reduce the high dimension from a very high dimension, could be thousands of cells, genes, and to a 2D, from high D to 2D. Okay. Any quick questions for this part? Now we move to the QC, okay, quality control of this matrix. If I have X matrix, so the first thing I want to know is what's the summation of this, okay? So each number is a count of the mRNA transcripts of that gene. So I want to see what's the total UMI for each cell. So I need to summate this to get a number to have some idea of whether this cell has been sequenced deep enough. Assume they are similar size, so the total amount of each individual cell are almost the same, maybe some fluctuation. So we expect the total number of this summation should be the same. So the first column, you got 8, this 5, 6, 12, 10. Yeah, so we want to first know the total library size. So this is called a library size. Basically, the total amount, the count of the rates, the summation for each cell. So how can we compute that in the MATLAB? So we have, remember we have X is still the same. So you just do a sum and then X. So you get this number. You want this number to be across the cells to be similar. So if I have a really small outlier, that means, for example, this particular cell, this is not so different from those. Still in the allowed range, but if this is extremely small, I would see this number 2 cell has not been sequenced deep enough. Maybe it's a contamination. There's empty droplet there's no cell in there, but this is environment fragments from other cells, contamination, but the sequence still have some rates, but not high enough. So in that case, this particular column should be removed from the next step analysis. This is original, but we are going to remove one of the low coverage cell because the library size is too small. So this particular QC is something like this. We certainly can do by rows, and this particular gene, let's see, 8, so you compute 16. So in MATLAB, you do by 2, so this gives you the column. So 8, 16. Okay, so this is good. So in our case, there's no empty gene. So if you have a row, for example, gene number 3, for all the cells you sampled, there's no information. So for the next step, you want to remove this gene unless you have a very compelling reason you want to keep empty genes in there in your analysis. So you believe if you want to concatenate with, compare with another sample, you have X1 with this gene not expressed, but in X2, another sample, you have this gene highly expressed. So in that case, you want to keep this, okay? But in general, and this gene is lowly expressed, undetectable in your sample, in your so many cells. So in this case, this gene should be removed, and this cell, outlier cell, should be removed. So we need to know how to remove cells, how to remove rows, how to remove the columns. So let's remove, so we have X. So let's remove second cell first, then remove the third gene out of it. So we are going to make a shrinked X. It's as a step of our QC, okay? So to remove X, the second column is put this into nothing. So in this way, you remove the column first, okay? You remove this by some kind of a cutoff standard. So because I have this summary statistics, I know I should remove number 2 cells. Sometimes this higher outlier also have to be removed. If this is a 24, I would consider this a duplicate because 2 cells would be captured in the same droplet. So you doubled your amount. So in that case, this too high is not good, too low is not good. So we need to have some range. So this function, this operation will remove the second column. So now you should have that removed. So now it's gone. This is gone. So you get this. To remove the third gene, so the same kind of operation, but just make sure the index, the third gene, which rows, okay, you remove. Now you have a shrinked minor X. So from the original becomes this clear, this is the original. And now you have a clean X. That's all we need to do, all right? So in the program, so I have these procedures allow you to select, to look at your data and select your cutoff above which number you consider too high, below which number you consider too low. So we will set this up. And also this procedure to remove rows and columns will iterate, because after you remove the columns, the total number of the rays, this will change to this. Because if you remove this, if you remove this cell, and this gene become empty, because it was a number for that gene. So if you remove the cell, and this gene should be removed. So, but after you remove gene, some of cells also have to be removed. So we will do this several times until nothing we can remove, so we finish the QC. Okay. So we are going to use real data to import the published data to get this, which is much bigger than what I can write on the board here. But this is a small example, give you an idea. Okay. So now we are going to visit the page, the paper, the link in the channel. It's a study of porosis. So it's a skin sample. So what I can do, normally I just type search, then GO. Okay, GO will lead you to accession number. So they download some, whether they have. Yeah, so if you look at the data availability, so we are going to use the data they generated in this study. They generate data and put into this record. So if you click, you should be on this page. There are 20 samples. We just pick one of them. So let's just select one of these samples. You can have a control or lesion, doesn't matter. It's just use this as an example. So I'm just, by random, I select this particular sample. I will copy this sample ID. So sample ID is start with a GSM, a number. So this is sample. And for study, you have a GSE, so the number. So this is a study. One study may have multiple samples. What we need is a sample level accession number. So we always need some like GSM. So I copied this and come back to MATLAB. And use our AG2. And then I will select this GEO accession number and replace this with the one I want to import. So after downloading, I should have this big X matrix in the memory. So I will first very quickly to get rid of those genes with no expression, okay? So this is a detection of the gene name. There's some gene name are the duplicated. I just add underscore or you just remove them. So there are different ways to treat them. But normally it's a very small number of genes, so you don't worry about them. Well, this is a huge data set. There's 36,000 genes. Normal cell doesn't express that many genes at all. So I believe there are a lot of zeros in this by rows. So we are going to remove all the zeros to shrink the number of genes. So at least you have to one cell express that gene. So the place we can do is this is edit, then you have the place where in the edit, the first filter gene cells, I will remove okay, if you look at this, so eventually we can see like this short cutoff because everything has been set up by default. But I want you spend some time visit those options to have a more customized control for yourself. The first step, we can remove empty genes. What I just described, if you have all 0 rows, we just remove you, okay? Let's see how many genes we can remove if you remove empty genes. Yeah, remember we had 36 something, but now it's only 27,000. It's quite a lot. More than 10,000 cells are empty. We remove those genes, then you can also click this filter button here to achieve this. Now let's look at this diagnosis tool. So this will create a scatter plot, which I will count each cell's this library size, and with respect to the mitochondrial rates ratio. So mitochondrial rates are those UMI in the gene encoded in the mitochondrial genome. There are 13 mitochondrial genes encoded in the mitochondrial genome. So we don't want that number to be too high. That is because mitochondrial membrane is rigid. You're supposed not to be broken extensively. So you shouldn't release many mitochondrial gene expression in your cell. So if that ratio is too high, it's indication of the damage of the membrane. Some cell dies. So let's see this scatter plot. If you select that, you look at this scatter plot. X-axis is the library size. What is the library size? Library size is just total amount of those rays in each cell. So you can see some cells, like this one, have a really high library size, 70,000. So the number in this column, 70,000, will compare average maybe somewhere just 1,000, 10,000. This is 7,000. So this particular cell and this cell definitely is outlier. There's so many rates dropped in there with the same amount of gene. So they are definitely something that we need to remove. And so we need to set up a cutoff somewhere, say 50,000. Okay, I will drag this red bar to 50,000. Anything above that, I will consider them as a duplets. Okay, 2 or more cells have been captured in that droplet. So each point is a cell, okay? And we don't want them to be too low. If it's very low, that means this cell has not been sequenced deep enough. For the y-axis is mitochondrial rates ratio. So we don't want that ratio to be too high. If you have a 90% or from mitochondria, basically definitely tells you that this cell is damaged. There's nothing in there. There's all a bulk of mitochondrial DNA transcripts in there. So we will set this percentage to be 15% and set cutoff of the library size to a certain range. So you can set this. So for the x-axis, lowest we need, say, 5,000. So this number, library size, each cell has to be greater than 5,000. And the maximize, so maybe 50,000, okay? 50,000. So we have a range, 5,000 to 50,000. All the cells have that library size in the middle should be okay. So set this to 50. And the y is the cutoff, so it is 0 to 15%. So after we put that number, and we can say click. Okay. So this cutoff is set up, and then you know all these cells will be removed, and you will keep the cell which in this particular range defined by this 2D space. You have to not too high, not too low library size, and they need a small percentage of mitochondrial DNA transcripts in there. Okay. Then you select OK. Then the total number cell and the gene will change. Just remove all of them. Okay. Okay. Now I have a clean data. So this is much shrunk. Now I have left with 7,024,246 cells. So that's what we call clean cells. Okay, it was huge. Okay, definitely we cannot get that amount of numbers. There are other ways to do this. So this scatter plot, which have a library size versus number of genes. So you see, if you sequence a cell more, and you should see more genes expression. And you detect, you sequence it deeper, and you detect more genes expression. So you can see this positive correlation. So in this case, we don't want have those outliers. I don't know the reason, but the normal cells should not have extremely high number of gene detected but then you can set this cutoff if you want, then make it slightly cleaner. Okay, so in this case, I will remove 19 cells by this selection. There are other options. So if you, for example, if you look at the name of genes by the first button here, you see some of those this is an antisense transcripts of that particular gene. So we know this gene, and we can search, and they are opposite direction in the gene body. There's a antisense transcripts, so S1. It's informative, but at the end in the analysis, maybe we not be able to interpret what they're doing. So we need maybe to keep this gene and just remove those if we don't need that. There are many of them. Those genes, for example, they are not named gene, and even though at the end you found this gene from your analysis, you have no idea what they are doing. So for some quick study, you just want to remove those unnamed gene because you want to have some interpretable very quickly, get some results. As long as you're not really focus on those dark matters in the genome. So if you decide to use those only named genes, you can select, say, I will remove you can select D here, just remove those gene without approved symbol. So let's see, this is a human. That's human sample, yeah. Select human. This will remove 8,000 genes. It's gone, accept. Now if I look at the gene name, yeah, so it makes more sense somehow. You can remove by the patterns. For example, I will see, okay, I will remove this all the gene with AS antisense. We'll remove all the genes started with LINC, all the names with a GM, so this is for the mouse. And there are a lot of this unnamed gene. So if you do this, you will remove another set of the genes. So this makes this more condensed, informatively easy to interpret the results. So now, if you look at your gene list, I remove those antisense, so now it's much clearer. Okay, this is optional, so it depends on your problem. You may want to discuss with your PI. So the PI say, okay, no, we have to keep antisense gene in there because we may discover some significant difference between comparison cross samples. We need that. But most time we don't really need. Another problem is if you remove this gene, again this gene, and you have to recompute the library size. Because the library size after you remove so many genes changed quite a lot. There's some undetected or lower coverage cells will be showing up after you remove the genes. So you have to do several rounds of the QC. There are other options. For example, people know the MALA1. This is a non-long coding RNA, but it should be in most of the healthy cells. That's by the empirical evidence. If you don't have a MALA1 expressed, there's something wrong with the cell, but people don't know why. Okay, so this is a long non-coding RNA gene, and abundantly expressed in all kinds of tissue, all kinds of cell types. So I put that in there. There's paper support this people study the quality of the cells expression. So if we don't have MALA1 expressed, have you remove the cell? So I click this, only one cell is highlighted. So there's only one cell without that particular gene expressed. I can find it where it is. Yeah. So there's one cell in the middle of this. I don't know if you can see. There's one cell in the middle here. No MALA1 gene expressed in that cell. So something wrong with that cell. So I can remove it. Okay, in general, to make those steps easier, so you can have this selection, basic QC, which contains, if you use stringent, you have the library size cutoff, which the upper bound has to be, the lower bound has to be 1,000 at least, okay? And 15% of mitochondria or less are allowed. And you have to have a minimum non-zero cells per gene, 15, okay, number. Minimum non-zero genes per cell, so 500. We set those numbers up, and this QC procedure will go remove the cell, remove a gene, and do again, and until no gene, no cell will be able to remove, then we are done with the QC. So for the most case, we found this setup is okay, will not remove too many cells. But remember, cells by themselves should have a huge range of this library size. For example, macrophages will be much larger than T cell. So you expect some variation. Okay, so this is just computational setup for the cutoffs. You have to decide by yourself when you play with real data, okay? And we know in the heart, okay, cardiomyocytes, you expect higher number of this mitochondrial rates. So 15% may be too stringent. In that case, you want to 20% allowed or less allowed. So if you select this, then the procedure will iterate and then converge and stop and see, okay, there are 2,933 genes will be removed, and also 301 cells will be removed. So in this case, if you accept the change, you will get the clean data ready for the downstream based on this pre-select cutoff. Okay, so this is today what we are going to cover. And so, as I said, next step is normally just do the embedding. Okay, so embedding this, and then you use UMAP or T-SNE. So if you use UMAP, so the embedding is the same procedure as I described here. So you compute the distance matrix and then use multi-scaling to project the results in there. Okay, just reconstruct the map. The principle is here. The procedure is slightly different because we know there's a conventional multi-scaling dimensional scaling will not generate the beautiful results like this. This is a procedure called UMAP. It's better than this classical multi-scaling dimensional scaling. But the principle is almost the same. It's reduce the dimensions for the project the relationship between cells into a map, which 2D or 3D you can visualize. Keep the relative similarity, relative difference, distance between cells as they were in the original high dimensional space. Because each cell here is defined by how many genes. Depends on number of genes, that's the number of dimension, original dimension of that cell. So it was 14,000 dimensions. So it's really, really high dimension data. We projected back to 2D. That's the idea, okay? In order to understand their interrelationship, similarities between cells. Okay, that's all for this Thursday. Any questions? Okay.
I think I missed a step. How did you bring down the number of cells?
Bring down number of cells. I use this pre-set up this QC, so it's automatically.
Yes.
Yeah.
I did that.
Mm-hmm.
So over here, I clicked on edit first.
Yeah.
Initial steps.
Yeah. It's supposed to have that. Yeah. Then the user.
Done.
Yeah.
I also removed cells with
No. Mm-hmm.
expression.
Mm-hmm.
What else should be removed to bring the number of cells down?
You don't have to. You just do this. Right?
I did that.
Mm-hmm. We use different samples.
I think
Same sample.
Same sample.
Mm-hmm. You have more genes. Yeah. It's okay. It's okay. Yeah.
Okay.
Not much difference. You will get something similar. Yeah. Okay. See you next Tuesday. Have a good weekend, everybody. Okay.